So I would like again to thank the Nobel Committee, the Royal Swedish Academy, and mainly to you. I gave part of this lecture in May, but with here, not in this building, but with a little different perspective. So first I will give a short introduction of what type of question we ask. Then I will try to give kind of historical perspective and focus on two types of problems that we have been exploring. So it will move sometimes simple, sometimes less. Now, in biology, there are very complex systems like signal transduction in the cell. Somebody could look at this and say that this is hopeless, at least if you come from more physical direction. But if you try to focus on individual junctions, they are just proteins, I say just, and there is a lot of information about proteins, particularly now structural information. From a very humble humble beginning at the late 50s, where both Perutz and Kendrew solved hemoglobin and myoglobin. Then there was advance, which to me is very interesting, solving enzymes by D.C. Phillips and David Blow. And then a stage where a lot of proteins could be solved, sometimes with robots. And you could try to ask, or continue to ask, what they are doing. Now, my main interest have been for a long time with enzymes, and I ran this stuff to my granddaughter, so this is how you explain it. Enzymes chop bonds much faster than the same bond without the enzyme, sometimes 20 orders of magnitude. So we have here several questions. One, how it is possible to be so much faster, which I think is still extremely fascinating. The other, understanding it, could you do something about it? And when it goes to understanding, a perspective change with time, but since we have such a complex system, such a large system, and at the end of the day you want to understand energy, you do need a computer. You cannot send a small person to push here the atoms. You get the structure from crystallography or NMR, and you try to see how it works. Now, a way to see it is when you see a clock, you could see how it rotates. Still, to some of us, it's not clear how it is working. And one way to see it is that biochemists discover the clocks, crystallographers show us all the parts, and single molecule spectroscopy now could show us how fast it moves. You still want to be able to understand how it works and to model it. Now, my introduction to it, I was not grown up on science, though as a young person I did some experiments. Don't take it personally, but I did parachute cuts from a very low altitude. I think that Nernst's dad did the same. And also tried to build guns, but nothing particularly until I went to the Technion, where I did experimental work on the speed of chymotrypsin, trying to measure very fast reactions, and concluded incorrectly that enzymes do not work by electrostatic effects. Because if you change the screening by salt, it appeared not to do much to the speed of the reaction. I also was introduced to physics course Very, very complicated course, but what I got from it is that many times, if you know the results before the reaction and after the reaction, you could figure out what happened in the real active region. And I essentially told my friend that one day I will write a symptotic wave function for an enzyme. Of course, I have no clue what exactly I'm talking about, but essentially I will show you that this happened. After the Technion, I read an Israeli article in the newspaper about Shneur Lifson, and he just became the scientific manager of the Weizmann Institute. But what attracted me to him was that his kibbutz near David was about 3 kilometers from mine, though he left this kibbutz long ago. I joined him, and we produce, especially when Michael comes, several landmark papers in molecular mechanics, basically developing approaches that allow you to treat any system as ball and springs, which allow you to minimize proteins, to ask a lot of questions, and it's also the foundation of what is now molecular dynamics of proteins. So I'm just saying that this was done. And near the end of the PhD, I was not sure what I'm really going to do, so I collaborate with a friend and describe a reaction as a combination of quantum mechanics and molecular mechanics using valence bond. And we could explain very, very large isotope effect. Then I joined Martin Karplus for a postdoc where we also use QM plus MM, but now in a simpler way, looking on the pi electrons of molecules like retinal, describing the pi electrons quantum mechanically, and the rest molecular mechanics. I was still interested in enzymes, so when I returned to the Weizmann Institute, I decided to try to use the same approach for enzymes. It looks straightforward. I just have to write a program that treats all the electrons quantum mechanically in the active part. Michael at that time was studying lysozyme strain effect, so I took his program and basically data, put it with my program, and the enzyme never dreamt to work. We could not break any bond. It appears that if you just connect bonds as ball and spring and treat part of them quantum mechanically, you will never understand enzyme catalysis. You will also not understand many other things. And the main missing part was the fact that when you break a bond, the environment stabilizes the charges of the broken bond, and this appeared to be the key factors of proteins in doing chemistry in biological systems. Now, as in many cases, it involved only 2 lines in very big computer programs, except that nobody else knew where to put these lines. And it resulted in this paper, this 1976 paper. And both this paper and the folding one were basically unquoted for a very long time. Now, this approach of having part classically and part quantum mechanically is a general approach where you focus on part of the system and put less effort in the surrounding. So this is called now multi-scale. Could call it a lot of names. It's also coarse grain. And it appears that if you really want to understand enzyme, not now, but maybe 30 years ago, it was better not to use molecular orbitals, as you saw before, but to use valence bond which are really these asymptotic states. You describe the reactant as a force field, the product as a force field. You mix them to get reality, but you get reality not in the enzyme. You learn everything in a reference system. You do not try to invent chemistry in your computer if you try to understand enzyme. You learn the chemistry in water, and then the only part that you do is moving from water to the protein by changing the surrounding. This approach appears to be very powerful. Here, for example, with GTPase, RAS gap complex, you could produce nice movies where this water molecule attack the phosphate and break the phosphate-oxygen bond. This is how you control signaling in biology. A lot of systems work like this. It's also the same reaction which work at ATP when you release energy. We will talk about it. Doing the movie is the simple part. Much harder is to get meaningful numbers by doing free energy calculations. And essentially, one of Johan Åquist's greatest contributions was to tell me or to show that if you run this longer, you eventually get the right result. Because at the early time, I always have my doubt whether this will work or not. So you need stronger computers. We also use it very extensively to study mutations of different residues. It works extremely well today. These are Christmas mutants, where one of these is calculated, one is observed. And this approach became very effective in studying, in enzyme design, in other questions, and eventually, in the last 2 years, finally, you could also do it with real ab initio QMMM and free energy calculation. A lot of people, this is phosphate hydrolysis, this is the quantum part, this is the classical. This is really hardcore supercomputer simulation. And again, we do not recommend to do it in the protein, except to impress the wrong people. You do it in water, you get conclusions about what is happening in water, and then you go to the protein using the EVB. But sooner or later, this will be done routinely, also on proteins, once people understand how to assess what they have. We also solve, in my opinion, the puzzle how enzymes work. Usually it's a long lecture, so just a few comments. In water, you have to spend a lot of energy rotating the water to stabilize the transition state charges. In the protein, you don't have to do it. And this energy, which is the energy inside the protein, it is not the interaction between the protein to the substrate. It's the interaction between the protein to itself, which is the secret to enzyme catalysis, and this is why it was not solved before computers, because it is not where you're looking for it. We also produce many papers, most of them, on how enzymes actually do not work. I enjoy much more to talk about it, but we don't have time. So now we move to the second half. This is for enzymes and also for short reactions. And when you have short reactions, you could run regular Newtonian dynamics on all the atoms and figure out what happened in few picoseconds, and what you will do will be correct. For example, this is an early movie of the vision process, the first step in the vision process, done in animation studio in the 1980. Everybody was fired already from this long ago, from this institution, not the National Eye Institute, the Hanna-Barbera. And what you have this model of retinal inside active site, light cam. This is taken from actual simulation. Molecule moves in the excited state and creates strain in the protein. You could see the same looking on the energetics. I will show you for 2 seconds. It goes relatively slowly in real time. So you could do the same thing with the potential surface. It's on our website, so I will skip it. The main point is that it did correct predictions very early on because it was a very short reaction and because the protein only introduced a constraint. We use similar approach in photosynthesis. We just got a guy who got prize, which is kind of related to photosynthesis. And now we go to the second part, what you do when the reaction takes very long time. And when you have very large system. So you want to know what happened? We use coarse grain approach, which started with this work with Michael, where we replaced the side chain by sphere. This is another paper whose quotation was originally extremely slow. And I always enjoy it because I thought that my paper are not quoted because I don't know how to write. But this was Michael and also was not quoted. Anyhow, now it's going up. And what we did, we taught this model to understand electrostatics. And then it started to do, I would call it miracles in understanding large protein function. So I will show you a few examples. First, when we do it, we also want to model very long time processes. So we don't want to do it by direct molecular dynamics, we want to do it by Brownian dynamics. You see, these fish feel all the forces around, and you have to find the right friction to do the simulation. And we invented a way to do it by taking the whole protein, applying on it very strong force, so it could move in short time, and then taking a small system, two-dimensional model, with friction, training both models to give the same speed under force, allow us to use the simple one without force, and to do very long time simulation. We could simulate milliseconds, sometimes even seconds. So show you several examples with this. ATPase. It's this molecular motor, which was solved by Walker, this Boyer, the other got Nobel Prize for figuring out how it works. There is a general idea how it works, but moving from structure to the actual function has not been done before correctly. There are many incorrect works. And the question is how rotating of this central shaft change the structure of these units and lead to chemistry. There are beautiful single molecule experiments that tell you something really funny. This supposed to have it's like 3-cylinder engine, but instead of rotating 120 degrees each step, it rotates 80, rest a little, does the chemistry, and then continue. The question is, could you understand how it happened without telling it that it happens, which is what most people would do? Could you use the structure? So we took the structure, this direction is the rotation of the central shaft. This is the change of structure of the subunits. And first we produce, without telling the model anything, that at 80 degrees it starts to be difficult to rotate. And then combining it with the chemistry, we got this extremely interesting diagram that if you spend 1 day on it, you will understand much more than any textbook ever told you about how ATPase works. I don't have the time, so I recommend that you will do it. Everybody who sends me a good explanation will get 2 kronas in the mail. I have to, I don't want to become poor now. Anyhow, you go from high energy to low energy. This is the direction of chemistry. This is the rotation direction. And using this type of simplified model, but with electrostatic, you could ask these questions. We've done similar questions asking how the lower part work according to the Peter Mitchell hypothesis when protons are moving up and lead to this rotation. It's a nice story, but a little long. Again, if you work on F0, just understanding, if you are trying to understand from structure, that unless you generated a model that give you the right direction, you actually done nothing, regardless of what, regardless if you publish it in Nature. Now, the one which will explain most this challenge of explaining how something works is this myosin 5, which work in one direction. There are a lot of such beautiful movies. This is even not our movie. But the real question is the following. Okay, usually we'll have to go out. Usually, when you walk like this or like this, you walk in the same speed. In the case of myosin 5, this is not what happened. Okay. This was done by my young PhD student who is computer scientist, and I'm insisting that he will take chemical physics. So, but he's good at this. So we took the structure and focusing on the electrostatic actually found out that having a bent knee, you have lower energy than straight knee. This energy is actually exactly the experimental energy that one, just by luck of course, that one gets from force experiments. And when you put all of this in extremely complicated diagram, because this is not a game, you have to spend 3 months reading about the experiments, you find out that when you go forward, the barriers are much lower than when you try to go backward. And you could check it by doing mutations, by applying different forces. There is a lot of work to do, but this is how it goes. How much time I have? Okay, nobody wants. Anyhow, let's assume 5 minutes. So we use the same approach for asking different questions like ion channels. There are the effect of voltage activation, where when there is one polarity, potassium does not pass. In the other polarity, potassium pass. And while there are beautiful work on the flow of the ions under zero potential or some potentials, again, Johan did really beautiful pioneering work. If you try to model the electrode effects, and I was told by Chris Miller that asking about the electrode is actually a taboo in the channel community. Where I'm coming from, I cannot do anything unless I have everything in. So we have to put the salt, the electrolyte, and then you start to get nice stories. like for 1 second, you could simulate current, produce nice movie. We will couple it eventually with the voltage. The last story, very short one, again, something which is dear to people here. When you have ribosome, there are nice experiments of von Heine that when you pass peptide, grow a peptide in the ribosome, some of these peptides are not going through. They get stalled. But if you connect it to the translocon, which is the machinery that divides it between the membrane to the outer side, then it helps to pull bad peptides. So again, you could simulate it. Again, simulation very similar to Johan, but we did it with good and bad peptides and combine everything together, EVB, Brownian dynamics, membrane potentials, and got this beautiful picture, which might even be correct, where the peptide goes out. It gets confused here. Essentially, there are new Nature paper which shows that there are cases where the peptide got here. Then it had to become an helix, which we did not taught him yet, but the rest is all based on the calculation. When it go in some lengths, it's being pulled by the translocon. In other lengths, it is not. And it's very hard to figure out why it happened without such simulation. So simulations are very powerful. I will kind of conclude saying that you could use it for what people want us to do, like studying drug resistance, figuring out what will be the next move of the virus by combining the knowledge that the virus wants to do its chemistry, but you also want to unbind the drug. And my regular conclusion with the bunny, where is it? That the secret to structure and function is electrostatic. Wait, we show him again. Okay, refused anyhow. There were a lot of people who made very significant contribution here, but we like to acknowledge two locals. Johan, when he still was handsome, and Lynn in a relatively bad picture. And last but absolutely not least is my wife, who supported me a lot. Now she enjoyed the Prince company. What could we do? But thank you very much.
Thank you very much.
Thank you.